So, good afternoon, ladies and gentlemen. I'm Alexandra Hiscox. As has already been introduced, I'm a postdoctoral researcher here at the Laboratory for Entomology. Over the next 10 minutes, I'm going to be talking to you about the research that we've been doing into the development of odor-baited mosquito traps for malaria control. So I'm going to start with some good news. Over the past 15 years, we've seen great progress in the control of malaria. This map shows the prevalence of malaria among children aged 2 to 10 years in the year 2000. If we fast forward to the year 2015, you can see how much those red and orange areas indicating high prevalence have declined. This is great news. This is largely due to the use of insecticide-treated bed nets, indoor residual spraying with insecticides, and also drugs to treat the malaria parasite. But this isn't enough. We want to see this map looking light grey all over, indicating that malaria has been eliminated. Over the past few years, unfortunately, we're beginning to see a slowing down in our progress towards elimination. And this is largely due to resistance of parasites to drugs. It's also due to resistance of mosquitoes to the insecticides that we use to control them. So here at Wageningen, we're working on new tools for the control of malaria vectors that are not reliant on the use of insecticides. So I'm going to begin our story in the lab with the fundamental question of how does a mosquito find her blood meal? Human beings are constantly producing odours, and it's these odours which attract the female mosquito to fly towards her host and find her blood. Now, it's been identified by our group that some people really are more attractive than others to mosquitoes. And we've been able to identify which chemicals these attractive individuals produce that make them so attractive. By combining these chemicals into synthetic odour blends, we're able to mimic the odour of a real human being. So what do we do with that knowledge? We want to apply this in a way that will allow us to develop ways of controlling malaria. When a person who's infected by malaria is bitten by a mosquito, this mosquito transmits the parasites to an uninfected individual, and this creates an infection. Imagine the situation whereby we can use our attractive odour and put this in a trap. Now, the mosquitoes, rather than being lured to the people, are lured to the trap, and this removes them from the system. Gradually, this leads to the breakdown of the transmission cycle and elimination of malaria. So I'm now going to take us on a journey. We're going away from Orion, away from Wageningen, and away from the Netherlands. We're even going past my parents' house in the United Kingdom. And we're going south towards the equator. We're going to Kenya and the shores of Lake Victoria, and Wageningen Island. Wageningen Island is an area which is still endemic for malaria, and this is despite good usage of bed nets and treatment with drugs. This makes it an ideal location to test a new intervention for malaria control. So together with our colleagues at the International Centre of Insect Physiology and Ecology, we've been working on a trial to evaluate these odour-baited mosquito traps for malaria control. Our traps require a source of electricity, and that electricity is used to power a fan inside the trap. When this fan is rotating, it blows out the attractive odour, and that draws in the mosquitoes. Once the mosquitoes approach the odour, they're sucked inside the trap. And once they're inside, they can't escape, and they die due to a lack of food and water. So this doesn't rely on insecticides, but it does require power. And Wageningen Island is an area where very few houses have got a connection to the mains electricity supply. So we designed a system whereby these traps can be powered with solar energy. Along with the solar energy that's used to power the traps, we also provide a source of indoor lighting and mobile phone charging for the house. We call this system the Solar-Powered Mosquito Trapping System, or SMOTS. So now that we have an odour bait, and we have a trap, and we have a system for powering the trap, we needed a study design that would allow us to actually evaluate whether this intervention could be effective for malaria control. We wanted to be able to compare how much malaria there was in people with the trapping systems and how much malaria there was in people without. But we also wanted to maximise the effect of our intervention by providing the trapping systems to the whole of Wageningen Island. So this figure shows Wageningen Island. The gap in the middle is a hill where nobody's living. We divided the population of 4,000 households into clusters of 50 households. Beginning in 2013 and ending at the middle of 2015, week by week, we gradually provided these trapping systems to every single household on the island until we reached full coverage. This meant that at any given time point throughout the study, we could look at malaria in people without the traps and people with. And we could also look at how much malaria there was when we reached full coverage. Because this was going to take 2 years to gradually install all of the trapping systems, we needed a fair way of deciding what order we would do that in. So together with the community of Wageningen Island, we carried out a ballot. We put multiple different possible rollout sequences into a box, and all of these sequences were beginning in different areas of the island. And one of those sequences was chosen at random, and that's the one that you saw illustrated on the previous slide. So now we have a trap, we have a power system, and we have a study design called the Stepped Wedge Randomized Control Trial. The final stage in the development of our study was to identify outcome measures that would allow us to actually estimate whether we really were effective in controlling malaria. We took a transdisciplinary approach to this, working with a group of over 50 different people. We looked at all different aspects of the malaria transmission cycle. So we worked with industrial partners on the development of the traps and on the power system for them. We worked with parasitologists and entomologists to measure malaria and mosquito populations across the island in all 25,000 people and 4,000 households. We worked with software engineers who developed electronic questionnaires that could be conducted using touchscreen tablets in the field, and the data could be synced real-time and accessible from all of our partners. Mathematical modelers and epidemiologists brought together all of this data to allow us to estimate whether we really were effective in our aim of controlling malaria. Last, but by no means least, we also worked with social scientists. These social scientists carried out weekly community training workshops on the island so that the community were educated about malaria control and also about the new technology. Social scientists also carried out interviews, focus group discussions, and listening surveys, and this allowed us to measure the knowledge, attitudes, and practices of people to malaria control, existing methods and the new. So now the question that I'm sure you're all waiting for me to answer. After all of this effort, all of this research, did it actually work? These are the results for the mosquito populations. The vertical dashed line indicates the point at which we began the gradual installation of these trapping systems. So everything from week -40 to 0 was our baseline period. From week 0 onwards was when we were beginning to install the intervention. The dotted blue line running through the middle shows the average mosquito populations across the entire island. The red line at the top shows the populations of mosquitoes in households which had not yet received the trapping systems. And the green line at the bottom shows the mosquito populations in houses which had received the trapping systems. So overall, using the odor-baited traps, we were able to contribute to a 69% reduction in the populations of the main malaria vector on Wageningen Island. Great news. But was it enough to actually lead to control of malaria? It was. Again, the dashed vertical line shows the point at which we began installing the trapping systems. The dotted blue line running through the middle shows the mean population malaria across the whole island. The red line at the top shows the prevalence of malaria of people who were living in houses without the traps. And the green line at the bottom shows the prevalence of people who were living in houses who did have the traps. Overall, people living in houses with the odour-baited mosquito trapping systems had 30% less malaria than people living in houses which had not yet received them. So to conclude, yes, odour-baited mosquito trapping systems can be used for malaria control. We discovered that community engagement was an absolutely essential part of running a large-scale intervention study like this. We don't yet know whether the traps can be sufficient to actually lead to the elimination of malaria, and this requires longer-term follow-up of this kind of study. So what are we doing now? Well, we're very keen to know how solar-powered mosquito trapping systems could be incorporated into development projects. Could solar power, providing energy for traps and indoor lighting and mobile phone charging, lead to this idea of a healthy home environment? Could this intervention be effective against other mosquito species, such as the vectors of dengue or Zika or yellow fever? We'd also like to know whether we can make these traps even better. Could we combine them with repellents to push mosquitoes away from houses while pulling them into the traps? So on that note, I'd like to thank you all very much for your attention. I'd like to thank the community of Wageningen Island for their participation in this study, all of the fantastic Solar Mile team, and of course our funders through the University Fund Wageningen. Thank you very much.